All right, we're in chapter 14. We're going to talk about Mendel and his peas and the idea of a gene. So we were talking in our last chapter about meiosis and this idea of inheritance. And people have known for thousands of years that traits are inherited by offspring from their parents. But what scientists start trying to figure out is how does that work, right? And so two hypotheses, two educated guesses sort of arose. And these are the blending hypothesis, this idea that genetic material from two parents kind of blends together like yellow and blue paint becoming green, right? And the particulate hypothesis, which is the idea that parents pass on discrete heritable units, which we would now call genes, right? So it would look something like that. The blending hypothesis versus the particulate hypothesis. And if you think about it, you're looking at this family, right? And you're looking at these kids and you're going, yeah, they are a mixture of these two parents. And the question is, right, is it blue and yellow make green? Or do the offspring keep those discrete heritable units? Okay, so along comes Mendel, and fortunately, he was able to decipher a lot of this by studying pea plants. So he discovered the basic principles of heredity by breeding these simple garden peas. He just had really carefully planned experiments. So he was able to figure out some of the principles of inheritance that people had been wondering about, right, for many, many years. And so basically he was studying these heritable features that vary among different individuals, things like flower color, and this is called a character. And each variant for a character, such as purple or white for flowers, is called a trait. So you have the purple trait or the white trait. Peas were available to Mendel in many different varieties, and so he was able to examine lots of different characters and lots of different traits there. Now, I don't want to make it sound like he totally lucked out, like he stumbled on peas. He very well may have chosen this on purpose. And scientists still do this today, right? You pick an organism that's going to be easy to work with and help you actually answer your question. Humans are terrible test subjects, right? If he had tried to figure out inheritance by using people, it probably would have been unethical, especially for a monk, and it would have taken a really long time. So some of the advantages of using peas, like this was a really good choice. They have a short generation time. They have large numbers of offspring, so you can count them. The mating can be controlled. You can let plants self-pollinate, or you can actually cover the flowers and cross-pollinate them yourselves. Right. So Mendel starts tracking characters, remember flower color or the shape of the seeds, things like that. So he's choosing characters that only occurred in two alternate forms, right? They're either purple or they're white. They're tall or they're short. They're smooth or they're wrinkly. Okay. And he also started with varieties that were true breeding. And this means plants that produce offspring of the same variety when they self-pollinate. So purple flowers self-pollinating with other purple flowers, they are purple. Okay. And so what these experiments end up looking like, right, is that Mendel is mating two contrasting true breeding varieties, right? So he's taking a purple flower and mating it with a white flowers. And these are called the parental generation. That's the true breeding parental generation. The offspring then are these hybrids, and that's called it's called the first filial generation. We just call it the F1, right? So filial F1 generation. And then of course, if you take the F1 generation and cross it with the F1 generation, that gives you the F2 generation. So you're going to see this terminology, right? We're going to have to work through this. And this is kind of what Mendel sees, right? So he takes those true breeding plants, a purple and a white, and he crosses them. Okay. And in that F1 generation, all the plants have purple flowers. But then when he crosses that next generation, right, pollinates F1s with F1s, regardless of whether that's self-pollination or cross-pollination, in the F2 generation, most of the flowers are purple, but some of them are white. And in fact, it actually comes out to this 3-to-1 ratio. So this is a lot of plants that he's counting, right? 705 were purple, 224 of them are white. It comes out around this 3-to-1 ratio. And so basically what he starts figuring out is that in the F1 generation, it's only that what he's calling like the purple flower factor is what's affecting the flower color in the F1 generation. So he actually starts referring to that as the dominant trait. So in the F1 generation, for whatever reason, this purple is what is always appearing. So that's the dominant trait, and white is the recessive trait. And it's not like that recessive trait, like the white factor, right, was diluted or destroyed, because it reappears in the F2 generation. Now, I know, this is so, like, it's really hard in biology, right? Because this is kind of like a history lesson, and you're supposed to be like, whoa, go Mendel. But if you try to envision what he's figuring out, based on pea plants, like if you can kind of put yourself in his shoes, he doesn't know anything about DNA. He's just a monk with a garden. This is pretty incredible. So here's a further look at some of the data he collected. So he did not just study flower color, right? He actually observed this same pattern of inheritance in other pea plant characters. So again, he was only studying ones that had two traits, right? So the seed color, yellow versus green, shape, round versus wrinkled, right? Is it tall? Is it short? And so what he's seeing, what he called these heritable factors, is what we now call genes. So notice that in all of these, flower color, seed shape, stem length, this is coming out to that same 3-to-1 ratio. And notice, this guy had some time on his hands. Imagine what you could do if you didn't have a cell phone, right? And you were actually bored. You could count thousands of pea seeds, right? He counted 6,022 yellow pea seeds to 2,001 yellow. This is a 3-to-1 ratio. Okay, so he's seeing this 3-to-1 ratio over and over and over again, and you gotta go, well, that's weird. So here's what Mendel comes up with. So he basically comes up with 4 discoveries, right? So what he's going to say is, first, right, these alternative versions of genes, right, or heritable factors, as he would have been calling them, account for the variations in these inherited characters in flower color or seed shape, right? And then for each of those characters, flower color, an organism must inherit 2 alleles, 2 different versions of the gene, one from each parent. Again, we don't know about DNA at this point. Okay. Third, if the 2 alleles differ, right, those 2 that you just inherited from your parents, then one of them, the dominant allele, determines the organism's appearance. The other, the recessive allele, doesn't show up at all. Okay. And fourth, and this one is super important, is this law of segregation. And this informs our understanding of meiosis. This idea that when there are 2 alleles, right, they actually separate during gamete formation, during meiosis, and end up in different gametes. Remember? So you had like the homologous chromosomes, they split, and then the chromosomes split again, the sister chromatids split. And so this is that law of segregation. So if a picture helps, right, this is where we're talking about alternative versions of a gene. So what we're showing you here, right, this is the chromosome you inherited from dad. This is the chromosome from mom, right? Notice they're referring to this as a homologous chromosome. The locus, plural is loci, this is like a location along the chromosome where an actual gene is located. And so they're in the same position, but notice the gene at this locus differs. From dad, we inherited purple. From mom, we inherited white. And notice they're even going down to showing you at the DNA level, this can be as simple as a single change, right, in the nucleic acid, right? Having a C instead of an A in your DNA code might determine whether or not you produce a protein Here we have an enzyme that's actually going to give us this purple pigmentation. And we'll get more into gene expression and what that means and where this protein is coming from, right? But this is the idea, is that a gene codes for something. In this case, it's a pigment. Okay, so again, this is a lot for Mendel to figure out without knowing anything about DNA, without knowing anything about chromosomes. He's just making really careful, detailed observations. Okay, and so as you start to think then about that exact same experiment we saw Mendel do earlier, he's saying, okay, well, purple flowers, and these are the true breeding, right, the parental generation, those have 2 copies of this dominant gene. And I'm actually going to change this picture, right? So when we think about what ends up in the gametes, right, you have the possibility, right, if you're a purple flower, of either giving a capital P or a capital P. We use capital letters to distinguish dominant genes and lowercase to point out recessive. And the P is a terrible letter, right? Like, how do you show that that's lowercase? I'll make it cursive. By the way, you can use whatever letter you want. So if they give you something like P and you'd rather use an A because you can easily tell the difference between capital and lowercase, go for it. Okay, right? So we're taking this jump now and saying, hey, that trait, that gene has to end up in the gametes, right? And we're getting different versions of that gene in the gamete. So basically then we can see what's really going on in that F1 generation. The physical appearance, what we call the phenotype, is purple, but the genotype, right, the actual gene organization, wow, that didn't work, is this mixture, 1 dominant and 1 recessive, and that is the only way where this F2 generation makes sense, where this white flower shows up again. So do you guys remember Punnett squares? I'm hoping that you guys have done these in the past, but maybe it's been a while. So just remember, you're basically just saying, okay, we're crossing a heterozygous purple flower, one that has a capital P, right, and also one that has a lowercase p, the recessive gene, with another dominant and a recessive, right? This is the sperm from one of the F1s. This is the eggs from one of the F1s. And then you pretty much just bring these into the middle, right? So 2 capital P's. We take a capital P and a lowercase p, right? We can see the genotype, the phenotype, the physical appearance is still purple. Same thing here, capital P, lowercase p, and then finally, in this bottom corner, we get 2 lowercase p's, and if there's not a dominant trait in the way, then we can actually see that recessive allele again. Okay. So Punnett squares, we're actually going to do some of these in lab. So think back to that if it's been a while. All right, so this is what it ends up looking like if you need it not in a Punnett square. And again, this is a good place to make sure we know the difference between phenotype and genotype. So genotype is literally which combination of alleles did you get. If they're the same, it's referred to as homozygous, right? Homo, same. I would actually go so far as to say, well, this individual is homozygous dominant. I can't write well enough here, but right, homozygous, I'll just abbreviate, DOM, homozygous dominant, terrible, right? Because he has 2 copies of the dominant allele. If you have the mixture, 1 dominant and 1 recessive, right, opposites, heterozygous, okay? So we got 2 of those. And then if you get 2 copies, again, that's homozygous, this white flower would actually be homozygous recessive. I'll just abbreviate again, homozygous recessive, right? So the genotype, if you look at the ratio there, you have 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. This is the same thing that we just saw here, right? The exact same cross. In the phenotype, and again, genotype is the actual combination of alleles. Phenotype is the physical trait, right? In this case, the color of the flower, this is where we see that 3-to-1 ratio. It's actually in the phenotype. Keep these two in mind, right? We're going to work through some genetics problems, some Punnett squares, and you'll find a lot of them are asking you to tell you what the genotypic ratio is or the phenotypic ratio is. And so make sure you know which is which, so you get those questions correct. All right. Yep. So it's not fun. One of the things you can do is what's called a test cross. So if you have a purple flower, so the cool thing with having the white flower, the cool thing with having the recessive trait show up in the phenotype is you know the genotype, right? The only way to get a white flower is 2 recessive traits. The dominant phenotype, the purple flower, you don't know. Is it a homozygous dominant plant or a heterozygote? And so this is kind of a neat way to do this. They're setting up basically 2 predictions, 2 possible Punnett squares. If, right, it's homozygous dominant and you cross it with a white flower, all of the offspring would be purple, right? That dominant trait is going to show up everywhere. But if that purple flower is actually a heterozygote, that means it's got a hidden recessive trait and 50% of your flowers are going to be white. So that's what they refer to as a test cross. If any of the offspring have the recessive phenotype, then it's the mystery plant there has to be heterozygous. Okay. Now, this can get more complicated, and it will for us, right? So a lot of like that purple flower experiment that we're looking at is what's called a monohybrid cross, where you just have a single character that's being crossed, right? So we're looking at flower color, monohybrid cross. But in order to kind of pull out some of these more complicated features of inheritance, Mendel actually did what's called a dihybrid cross, where you actually use 2 true breeding plants for 2 different characteristics, right? These are our dihybrids. So notice what we have here. In our parental generation, right, true breeding plants, we have a plant that produces yellow round seeds. Those are both dominant traits. And we're going to cross it with another true breeding plant that always has these green wrinkled seeds. Okay, so the only gametes that this guy can pass on are yellow and round, and the only gametes that this guy can pass on are green and wrinkled. Notice they don't change the letter to like a G on this side. That is the recessive version there of what we call of the color. Okay, just like we didn't with the purple and white flowers, we didn't make it a W. It was just a lowercase p for a white flower. Okay, so in our F1 generation, we are going to get heterozygous for both of those traits. Now, again, we've got 2 predictions. We're trying to figure out, or Mendel really was trying to figure out, were those traits inherited together, right? Did they depend on each other? Were they going to the same gamete together? Or were each of the traits assorted independently, right? Are they spread out through the gametes completely independently of each other? So this hypothesis with our Punnett square basically says, well, if they have to go together, right, you'd always put the 2 dominant traits together, right? And the 2 recessive traits together. And so what you'd end up seeing is kind of this 3-to-1 ratio, right, of yellow round to green wrinkled. If they each assort independently. And so the way to think about this is you take, right, we're crossing this F1 generation. You're saying, well, what are all the possible ways that this could end up in the sperm, right? So in this case, we got the dominant trait for yellow and the dominant trait for shape, right? But we could also pick up the dominant trait for yellow and the recessive trait for shape, right? Or we could have gotten the recessive trait for, that would be green, I guess the recessive trait for color, the dominant trait for shape, or the recessive for both. And so and I don't have enough dexterity basically to do this, but when you try to draw these Punnett squares out yourself, you do the same thing. You say, what is every possible combination of color and shape in this dihybrid cross that could end up in a gamete, right? And so we do that on both sides, and we get this crazy Punnett square. Notice they're always keeping the two color letters together, and then they follow with the two shape letters. So even down here, right, where it's recessive, or this one's interesting, right? They brought yellow, or sorry, I guess that's green, lowercase y, lowercase y. That's why this seed is green. And then we said, oh, here's the dominant trait for shape, the smooth shape, and here's a recessive trait for shape, but that's going to be hidden. So we have recessive color and we have dominant shape. So notice how they bring those letters in. If you kind of always keep them in order, it's much easier to figure out this ratio. Because sure enough, what happens is 9 out of the 16 are yellow and smooth, 3 out of the 16 are green and smooth, 3 out of the 16 are yellow and wrinkled, 1 out of the 16 is wrinkled and green, right? And that is exactly what Mendel found, was this 9:3:3:1 ratio. And again, he does this with a bunch of dihybrid crosses, really meticulous record keeping, thousands of plants being counted over and over and over again. He's seeing this 9:3:3:1 ratio. And so that confirms his hypothesis of independent assortment. And again, the idea here is just that when characters, when those genes, what we call them now, right, are moving into gametes, it's totally independent of other different characters, right? So seed color and seed shape are not connected to each other in any way. Okay. We'll actually start the lab, so you can look through this both in your lab and in the textbook, thinking about this as flipping coins, right, and where that's going to happen. There are some exceptions to it. So Mendel did kind of luck out in the traits that he picked. This only works on genes that are on different chromosomes or that are far apart on the same chromosome. So there are some traits, right? Some genes, some segments of DNA have to be close to each other, right? Those tend to be inherited together. But this idea of this law of segregation and independent assortment really follows the rules of probability. And so then that's going to actually, you can then mathematically make a lot of predictions about how traits are going to be inherited. I won't go into all that. Okay, so that is kind of classic Mendelian genetics, the things that Mendel figured out, and it applies to an awful lot of stuff. There are definitely exceptions to this as well, right? So there are different patterns of inheritance that don't fit that normal dominance pattern, that normal Mendelian genetics. So for example, there's what's referred to as incomplete dominance. So this is a time when red and white make pink, okay? And so it's not always one flower color is dominant over the other. Sometimes you have this incomplete dominance. But notice what happens. It still follows those rules that it's not like white disappears. If you go ahead and cross that F1 generation, you actually go back to having some red and some white and some pink. So it's still showing, it's not showing a blending hypothesis. It's still that particulate hypothesis. Okay, but there is incomplete dominance. That doesn't follow Mendel's rules. There's also times when it's not so straightforward. So if you think about human blood typing, it's not A or B. You can have A, you can have B, you can have AB, you can have O. And so this is something that Mendel did not try to decipher. So this would be an example where there are multiple alleles. It's not just a choice between two outcomes. And so this is kind of an interesting one. This comes up a lot, obviously, in like a human anatomy class. So we have our different blood types listed down here, A, B, AB, and O. And really, when you think about this blood type, like what makes someone have type A blood is if they have a certain shaped antigen on the surface of their cell. So this one's the A antigen, and that surface marker came from their genes, right? So they either inherited 2 copies of the allele to have that A, or they got 1 copy of that allele to produce the A, and then the recessive trait, 2 copies of the recessive trait is how you end up with O blood, right? So the genotype absolutely still is determining the phenotype here, but now we have multiple different versions. And so this doesn't fit perfectly with what Mendel decided either. You also run into situations, this is called epistasis, where one gene that affects the phenotype, so here we're looking at the colors of these Labrador retrievers, right, where one gene affects the phenotype of another due to the interaction of the gene products, right? So like, how do I say this? Expression of the gene at one locus, at one spot on the DNA is actually altering the phenotypic expression of a gene at a second location, right? So here's an example of this. This would also fit under polygenic inheritance. So there's actually 2 different genes that affect the pigment color in these dogs. And so 2 of these genes are actually interacting. Okay. So one is determining the color and one's determining whether that actually gets deposited in the fur. Sorry about my phone. Okay, so epistasis is when 2 separate genes are actually influencing each other, right? Are we making the pigment, and does that pigment get into the fur? Okay. Another polygenic inheritance, how's this? And I love this, a simplified model. So polygenic inheritance, again, means that there are multiple genes that affect this trait. So skin color, right? There are multiple genes that affect skin color, right? And then so in this case, there's this like additive effect. And so you could have 2 parents, you know, with kind of this middle of the road pigmentation, you know, something kind of in here-ish. And it would be very unlikely, but it could happen that they had a very white child, right? Or it would be unlikely, but it could happen, right, that they had a very dark skinned child. So when there's multiple genes involved, Mendel's rules, it gets much more complicated. Height, for example, there's over 180 genes that actually affect height. So you can't just make a Punnett square, right, and decide what your kids are going to look like. And then it gets even more complicated, right? So you guys are all familiar with this idea of nature versus nurture. And so by talking about genetics and inheritance, right, we can get into this very much like it's in your genotype that determines your phenotype. You inherited these traits, right? But this is an example where it's actually the pH of the soil that determines the flower color. So how about that? There's a very much a nurture example there. So these 2 plants essentially genetically are the same, but their phenotype is different based on their environment. And so that could happen as well, right? You can have the genes to be tall and then not have a balanced diet, not have enough amino acids, right? Have gone through a period of starvation when you were a child, whatever, and you might not hit that height that your genes actually coded for. So the point is, it's much more complicated, but what we'll focus on really are the Mendelian genetics, because we can kind of wrap our heads around that, and it really is useful. So in humans, we tend to make pedigrees. We don't tend to do breeding experiments where we purposefully cross individuals to see what will happen in the offspring, but instead we kind of think backwards, right? And so you can try this with your family if you want to look back and find pictures. Who has a widow's peak, right? Does your hair come down into this little point or not? And one thing I will point out here, right, is that notice that having no widow's peak, so kind of like a flat hairline, is recessive. And so this maybe is a good time to point out a dominant trait does not necessarily mean that it's more prevalent in a population. Right? It just means that if you have 1 copy of it, it's going to show up in your phenotype. So in this case, I think actually this no widow's peak is much more common, but it is in fact the recessive trait. Anyway, so these pedigrees, you'll see some of these. Males are in squares, females are circles. Males that have the trait, if you have the trait, it's a solid filled in shape. If it's empty, you don't have that. And so then you can, show crosses. Those are the grandparents. These are the 4 children that those grandparents had. This grandparent only had 2, right? And on and on. And so really what we'll find is there's an awful lot of traits in humans that really follow that kind of basic Mendelian pattern of inheritance. And a lot of our understanding of how we inherit traits from our parents really goes back to Mendel's work. And sometimes this can actually be super useful. So we'll look at a variety of different I'm actually doing this in pathophysiology right now. We're looking at inheritance of certain genetic diseases. So this would be an example. If you're talking about a disease, when you've got heterozygous parents like this, we would refer to them as carriers, right? So in a recessive disease, both of these parents have the normal phenotype, right? They don't know that they're carrying this recessive gene, but sure enough, in our Punnett square here, there's a 25% chance, a 1 in 4 chance that their offspring will inherit both copies, right? And develop this disease called albinism, where you don't produce pigments. So carrier is the term we would use, particularly if this is a disease state. So one of the things that happens here is if this is a recessively inherited disease, right, it's only a 1 in 4 chance. And what are the chances that these 2 parents both found each other with this recessive trait? What you'll find is that inbreeding is going to increase the chances that 2 parents will be the carriers of this trait. So a lot of societies have rules about like marrying, your siblings and things like that. In Colorado, I think you're actually allowed to marry your cousin. Other genetic diseases. So achondroplasia literally means without cartilage. So this is a form of dwarfism where basically you don't have the cartilage to let yourself get taller. And so they're showing us here the inheritance pattern where if you had this surgeon here who has achondroplasic dwarfism, right, it's a dominant gene for this disorder, right? If he mates with a woman of a normal stature, we can run that Punnett square and we can say, hey, it's a 50-50 chance. And this is the sort of thing that becomes important, is being able to understand and explain, right, if you're going to work, say, in a medical field, to people what the chances are of passing on something to your offspring. So we can have some fun. I know a lot of this is like boring old Mendel and his peas, but there's a lot of real world application to this as well. Okay, so I'm going to end it there. I did want to point out at the end of the chapter, they had some really nice summaries of this because I know there's a lot here, kind of different patterns of inheritance, including those crazy like polygenic inheritance and epistasis. And so check out that summary if you need to kind of go over this again. Okay.